First off, I'd like to thank the organizers of the conference for giving me the opportunity to present this work in this great conference. My name is Spyros, and I'm a PhD student at the MRC University of Glasgow Centre for Virus Research. And today we'll be talking about exploring the natural origins of SARS-CoV-2 through recombination signatures. And I'd like to start off acknowledging all the scientists that have sampled and sequenced all the coronavirus sequences that I'm using in this analysis, because obviously without them, this project wouldn't have been possible. And I will be very much following the kind of theme of this session, which seems to be recombination. So I will be explaining why recombination is crucial when looking into the evolutionary histories of coronaviruses. And then with that in mind, I will be explaining what the closest relatives to SARS-CoV-2 we know of are. And if we kind of overlay the geographic and the host information of these viruses onto the phylogenies we have, whether we get closer to finding out where SARS-CoV-2 came from. And then very briefly, I'm going to finish off with some very interesting recombination hotspot analysis and what it might mean. Right, so to start you off, I really like this short review by Graham and Barrick, and I should point out that this was published in 2010, so before MERS or SARS-CoV-2 emerged. It essentially tells the same story that we see at the moment with SARS-CoV-2, that recombination is really important in these coronaviruses, and it happens very often. And it can be used by these viruses to kind of change their phenotypes and potentially help in cross-species transmission. And if you've been following the literature of all the new bat viruses that are getting sequenced, you always see a similarity plot. And in the similarity plots, you see some dips and some peaks, meaning that some parts of the new coronavirus genomes are more similar to SARS-CoV-2, for example, than others, indicating that there is recombination taking place. And this is not only the case with SARS-related viruses, but has also been seen with MERS-like viruses. For example, this paper describes this new bat MERS-like coronavirus that has this deep sequence similarity near the spike. And it always strikes me how similar these seam plots are. So why is recombination important? So imagine two ancestral viruses, 1 and 2, they find themselves in the same cell in a mixed infection, and they kind of recombine. Now, if you take the progeny of these viruses and take the entire whole genome of these recombinant viruses, align it and make a phylogeny, the phylogeny you'll get out of it will actually not reflect the true evolutionary history of these viruses. What you need to do is determine where the recombination breakpoints are and split the genome by them, and then do phylogenetics for each one of these putatively non-recombinant regions. And in this case, you would see that for this bit, blue virus 2 is actually closer to green virus 3 than yellow virus 1, and kind of the other way around for the recombinant bit. So what we did is take the 78 available whole genomes of sarbecoviruses. This is just the subgenus of the SARS-related coronaviruses, and take two approaches to determine recombination. So first off, and this is work done with help of Darren Martin, we used RDP5, and we used all the modules, all the recombination modules and implemented in RDP5, to find all the minor sequences, recombinant sequences in this kind of 78 sarbecovirus whole genome alignment. And then by detecting all these more than 150 signals of recombination, we can take out the putatively recombinant bits of the alignment for each virus, and then create this non-recombinant whole genome alignment that only leaves the major parent in the sequence and replaces the minor with gaps. And if you do a phylogenetic reconstruction of that, you get this pattern here, where SARS-CoV-2 is right here, and you can see about 20 related viruses that we know of in this clade, which I like to call the nCoV clade. And then just point at the topology here, you get this distinct change in clade to this non-nCoV clade where SARS-CoV-1 is found in, and then kind of the outgroup with these two coronaviruses, one sampled in Kenya and one in Bulgaria. So then the second approach we took is try, more similar to what I just explained in the previous slide, try and determine fairly conservatively the kind of most important breakpoint sites for the entire whole genome alignment. We used GUARD for this, and we have 22 breakpoints that if you kind of overlay them on top of the ORF schematic, they fall kind of here. Then we split the alignment based on each one of these breakpoints, and we do maximum likelihood phylogenies for each one. So then what do we see out of these maximum likelihood phylogenies? And I'll start off with just by naming the closest ancestors, closest relatives. So RATG13 is one of the viruses that was kind of the first ones to be discovered early in the pandemic, followed by ARM102, which has kind of a cooler story because it has a recombinant bit around spike. And then two more viruses that got published very recently are RPY06 and PRC31. And I have to mention that all these four viruses have been sampled in horseshoe bats in Yunnan, China. So as I said, these are just the genome schematics where each region is split based on those 22 GUARD breakpoints. And this is just a kind of arbitrary metric here, where for each maximum likelihood phylogeny, you split the nCoV clade from the non-nCoV clade, and then within the nCoV clade, anything that's very close to SARS-CoV-2 will get a deep kind of fuchsia color, and anything that's far away but still within the nCoV clade will get a softer pink, and kind of the other way around for the non-nCoV clade. You can see that these viruses have some recombinant bits, but I will focus on the one of the regions where they all fall in the nCoV clade. This is region 5 here, one of the kind of longest spans. And if you do the phylogeny here and you kind of zoom in, you can see SARS-CoV-2 here, RATG13 here, and then these three viruses form this distinct clade that I like to call the Yunnan clade. And bear in mind here that this is kind of a sister clade to where the viral population of the proximal ancestor of SARS-CoV-2 would be. So these four viruses, close relatives, have been sampled in China, but we have two more viruses that have been sampled outside China, and these are RAX203, sampled in Thailand, and these two almost completely identical viruses sampled in Cambodia. Again, you can see that recombinant bit here. And overall, these kind of fall further away from SARS-CoV-2 in the phylogeny than the previously mentioned four viruses. But for some of the regions, for example, region 4 here, RAX203 actually clusters within the Yunnan clade. For example, region 10, the Cambodia viruses form this kind of topology. And I mean, here you could argue that SARS-CoV-2 is actually the recombinant one jumping out of here. But what I'm trying to show here is that there is a lot of evidence of recent recombination events between these viruses. So for each region, the topologies look quite different. And to put some dates on all of this, we used a dating model in BIS1 that is kind of described in the previous paper of ours to put some dates here. And this is region 5, which I mentioned before. So you can see that we estimate SARS-CoV-2 to have shared a common ancestor to the Yunnan clade about 40 years ago. And this is a relatively short or long time, depending on how you see it. You can kind of estimate some very recent dates for the Yunnan clade, meaning that these viruses shared a common ancestor fairly recently. And then I haven't mentioned yet these two viruses that we estimate shared a common ancestor to SARS-CoV-2 about 100 years ago. And these CoV-45 and 21 actually got sampled in Zhejiang, which is a province of China in the eastern, and so all the way across China from where Yunnan is. So now let's look at some maps and see if we can see how these kind of geographic ranges relate. So this is, again, the region 5 dated phylogeny, just with less collapsed clades. And you can see SARS-CoV-1 sits here. And some pangolin viruses, I'm not going to talk about them, but you can kind of ask me at the end, there's a cool story. And if you kind of plot all this information in a map, so every region, so provinces within China, countries outside China, and every region that's colored is where there is a sample in this kind of 78 sarbecoviruses. And for the pink hue regions, there has been an nCoV virus sampled, and the kind of the pinker, the closer that virus is to SARS-CoV-2. So starting off with Hubei here in central China, which is just very pink because SARS-CoV-2 emerged here in Wuhan. And then you can see Yunnan with these 4 viruses I mentioned, and all the way to the east, Zhejiang with these 2 viruses, and this third one that only has 1 recombinant bit. I'm not going to go into detail, it's an interesting story again. And then you can see Thailand here with RAX203 and Cambodia with these RSH-STT viruses here. And this kind of geographic range for these viruses that we estimate shared a common ancestor for some of the bits in the past 100 years or less is not trivial. So the distance between Zhejiang and Thailand, for example, is about 2,500 kilometers. So we see that there's this kind of ancestral movement across this massive geographic range in East Asia of these viruses. So there's no clear clustering of where these viruses have been sampled, but maybe there is some clustering with their hosts. So if we look at the closest viruses, this is actually not the case, because almost every one of these viruses has been sampled from a different species of horseshoe bats in the Rhinolophus genus. But there's some interesting things you can get out of the host ranges of these species, of these bat species. So to start off with the bats with the widest ranges, Pessilus, Aphanis, and Sinicus, these have geographic ranges that kind of span across the entire region that I just mentioned these coronaviruses seem to be moving through. So from like Zhejiang to Yunnan and then down the Malay Peninsula. And then for some of the viruses, for example, sampled in the Malay Peninsula, so Thailand, RAX203, and these Cambodia viruses, these are found in bat hosts that have a bit more restricted ranges. So Chameli is only found kind of here, so not in China, same with Echinatus. And then RM102 has been sampled in Rhinolophus malayanus, which is only found in Yunnan out of the entire country of China. So there's this clear picture here that the overlapping host ranges of these horseshoe bats are actually allowing the movement of these kind of related coronaviruses. So now you hopefully have a kind of a general picture of what the origins landscape looks like. So I'm going to move on to some really cool recombination hotspot analysis. And this is work in collaboration with Darren Martin and his team, Arne de Klerk and Renzo Lawrence. And as I mentioned in the very beginning, we kind of determined this less conservative method of determining kind of recombination signatures in these 78 viruses. And you can use with RDB5, and you can use this positions of the kind of recombination breakpoints across the alignment to determine whether there are hotspots or cold spots of recombination. And we've used 2 different methods. not going to go into details about the methods, but essentially, the shading here for both plots is the confidence intervals, with using a kind of permutation test, and the kind of lighter gray is 99% confidence interval in both cases, and anything above, any line above the light gray is determined to be a recombination hotspot, and anything thing kind of below is determined to be a recombination cold spot. And there's a few places here that you can look at, and this is kind of the operating frame schematic. So this long one is ORF1AB, and then you get spike here. But I'm going to draw your attention to a specific place across the genome. And these are kind of consistent hotspots right before the spike open reading frame. And also in this analysis, we also pick up kind of one downstream within the spike open reading frame. And also these very persistent cold spots that are exactly where the N-terminal domain and the receptor binding domain of the spike ORF is. So essentially, this knowing the functional importance of these regions in spike, this is really indicative of kind of selective recombination for that region to be switching between these coronaviruses through recombination. And so some form of antigenic shift. And this is important, linking back to the previous kind of things I showed, because for almost every one of the very close viruses that I've been talking about so far, there are recombinant bits in about these regions that I just showed. So right at the start or slightly before the start of spike. And for example, for region 16 here, this is the non-nCoV clade, so far away from where SARS-CoV-2 sits in the phylogeny. And you can see that RM102 and RAX203 share this recombinant bit that's quite closely related. And then for region 17, you can see that these four viruses called 45, 21, and PRC31 or PRY06 kind of jump out to this side of the phylogeny as well, close to here. And we kind of think that these patterns are indicative of, so it's not necessary that these are independent recombination events, but these patterns are slightly indicative of what we like to call recombination overprinting. So there was an ancestral recombination event with the non-nCoV clade, and then because these viruses circulate together, there were more subsequent recombination events between them that kind of mask the original recombinant bit, and you get these discrepancies between these viruses. Right. So the takeaway points are that there is very clear frequent recombination in the evolutionary history of these sarbecoviruses. The closest relatives of SARS-CoV-2 we know of is this Yunnan clade, but we estimate that these shared a common ancestor to SARS-CoV-2 about 40 years ago. So we're still lacking the proximal ancestor of SARS-CoV-2, and what we need to do is obviously sample. But again, there's no guarantee that that Yunnan, for example, is the place where this kind of viral population resides, and because of this pattern of easy movement across East Asia of these viruses, which seems to be facilitated by the overlapping ranges and quite wide ranges of the susceptible horseshoe bat populations. And finally, the clear hot and cold spot recombination evidence around the functional bit of the spike of the open reading frame is suggestive of kind of an antigenic shift mechanism taking place in these viruses. So to finish off, I'd like to thank everyone involved in this work. I'd like to thank my supervisors, Joseph Hughes and David Robertson, the labs I work with at the CBR, the Robertson Lab and the Wilson Lab. I'd like to thank Darren Martin and his team, Arne de Klerk and Ranger Lawrence, and Sergey Pond and Wei Xia and Xiaowei Zhang. And I'd like to thank you for your attention, and I'm more than happy to take any questions.